is Morgan Wirthlin.
Hi, Morgan. I'm in Portland with Claudio Mello, and it's kind of a neuroscience lab, but my background is more in evolutionary biology, dolphins and things like that. But now I'm more of an omics person, deeply embedded in bioinformatics, developing new algorithms to study like novel gene evolution, promoter gene regulation, blah, blah, blah. And my interest in mannequins actually started when that Hackett paper came out in 2008, showing that now parrots and songbirds are sister taxa. And so we got two vocal learning groups with mannequins weirdly wedged in between there, thought to be non-learners. And so my idea is, well, what if vocal learning is not this binary of you have it, you don't have it, and more of a stepwise evolution? I mean, it makes sense, right? You've got this complex behavior. It doesn't pop up overnight. So a lot of my work has been focused on what are the steps of evolving a new behavior in the brain on a genomic level. And so I'm using the Manakin. Our lab kind of specializes in these in situ hybridizations. We've got like an Allen brain atlas for the zebra finch, and I was part of the whole 50 genomes project recently, comparing human and songbird gene expression in some of these vocal output nuclei. But I'm now applying that to things like the Manakin to try to understand, what aspects of this expression, this circuitry, do they have? Do they have it? And I think they do. And part of my thesis work is going to be now understanding, looking at promoters and trying to figure out how are genes activated in some of these shared and not shared nuclei. And yeah, so if you have, especially if you know of interesting vocal behaviors in mannequins, definitely come talk to me. I'm very interested in kind of doing this comparative work to understand what's the variation in genetically in some of these different animals that could be reflected on a genomic level. Yeah.